Good afternoon, everyone, and thanks OMICS group for the invitation to share our research results. And today is the last day and last session, so I will try to do it quick. I know everyone is tired and wants to go home, and due to the weather forecast. And so the topic of my talk is that how we can use both nanotechnology and medicinal chemistry approach and blend them together to solve the issue of cancer therapeutics. So this is some statistics. The WHO has predicted that in 2030 there will be about 15 million new cancer cases, and in 2030 about more than 11 million people will die from cancer. So in order to address that issue, we need to know what are the problems of the current cancer therapy approaches. So most of the cancer drugs, they are highly toxic, and since they are highly toxic, and it causes undesired side effect. And many of the drugs, so for example, Taxol, docetaxel, they are water insoluble. Since they are water insoluble, to giving the means IV of the drugs, so people use other excipients are mixed with the cancer drug, and then they are given the IV. So those other compounds which are mixed with the cancer drugs, that causes side effects. And these cancer drugs, they go to the tumor site, but they also go to the other sides of the body. And you probably have seen cancer patients, then they lose their hair, and it affects the reproductive system. So cancer drugs go to the cancer site and also other sites. So the drug which goes to other sites, so we lose that drug. So it's a wastage of the drug. So that's because and the cancer patients have to take lots of higher doses of drugs, causing more side effects. And since the drugs are small molecules, so renal clearance occurs. And so in order to address this issue, nanotechnology, it can help. So this slide shows the relative size of the nanoparticles. You see that these are various nanoparticles: liposome, dendrimer, gold nanoshell, quantum dot, fullerene. So ideally, when the size of a particle is from 1 to 100, they claim it is a good nanoparticle. And this is the size of a tennis ball, 10 to the power 8 nanometer, and antibody has about 10 nanometer, the relative size. So in case of tumor, so this is a tumor, and this is the blood vessel which goes through the tumor. So this tumor vasculature, it has a characteristic features. So the vasculature inside the tumor, so this is leaky. So this is a leaky tumor vasculature. And when there is a nanoparticle carrying the drug, there are two approaches for the treatment. So one thing is that this nanoparticle or the nanocarrier, it's carrying the drug. So it's like a soccer ball, and inside the soccer ball there is the drug. So there are two approaches for targeting. One is passive targeting, another is active targeting. So in case of, I will go to the active targeting first. So in case of active targeting, so you see this is a nanoparticle, and on the surface, inside there is drug, and outside the surface there is a targeting ligand. So it can be an antibody, it can be a small molecule or peptide. So this is a cancer cell. So cancer cell on the surface, it can overexpress certain receptors. So for example, ovarian cancer, they have overexpressed the folate receptor. So if there is a folic acid as targeting ligand, then it will recognize the receptor and it will deliver the drug. It will undergo the receptor-mediated endocytosis and deliver the drug. So this is active targeting. But there is another targeting, which is passive targeting. So there is no targeting ligand on the surface of the nanoparticle. So it will enter into the tumor interstitium through the leaky vasculature. And since nanoparticles are bigger in size, So they remain in the blood circulation for a long time. And this effect is known as EPR effect, enhanced permeation and retention effect. And due to that reason, it remains in circulation for a long time. So this is passive targeting. So when it enters in the tumor interstitium, it releases the drug slowly to the tumor cells, and the cells die. So these two approach. In our case, we used a compound. So this is a flavonoid compound called luteolin, and this compound is found in many herbs and vegetables, and say it's found in celery, cilantro, bell pepper, asparagus, and other peanut roots, and many other green vegetables. So we picked this compound. So this compound has been shown by other groups that it has anti-inflammation and anti-cancer effects. So it showed anti-cancer effect against breast cancer, head and neck cancer, prostate cancer, skin cancer, cervical cancer. So the way it functions is that it triggers the cell death by activating apoptosis pathway, and it can also suppress the cell survival pathways. Okay, so now we formulated the nanoparticle. So we took a diblock copolymer of polylactic acid and polyethylene glycol ether with a molecular weight of 10,000, and the ratio of polylactic acid and polyethylene glycol is 50-50. So we formulated a micelle type nanoparticle. So the hydrophobic core remains inside the polylactic acid, and the hydrophobic luteolin, it goes inside the core. Just the soccer ball at the center, there is this hydrophobic luteolin, and on the surface there is water-soluble PEG. So this is the solution of the nanoparticle, and if we lyophilize it, we have powder, so we can resuspend this powder and get the solution. Now the characterization part. So here, this luteolin, which is hydrophobic water-insoluble powder, if I add it in water, so it precipitates. It does not undergo solution. But the nanoparticle, it undergoes solution. So we characterize by using different techniques. So this is transmission electron microscopy, and it shows that the average size of most of the nanoparticle is about 100 to 110 nanometer, and this is the DLS, dynamic light scattering. And we did the UVVs to see the lambda max of these phenyl rings, so more conjugation, higher lambda max, and less conjugation. So, to find the characteristics of the luteolin inside the nanoparticle. So initially we did in vitro assay. So this is the first one is the SRB assay, sulforhodamine B. So we took two different cell line. One is head and neck cancer cell line TU212, and another is lung cancer cell line H292. So in case of SRB assay, the cell growth inhibition, so we saw that the green colored one is that nanoparticle, Nano-Luteolin, and pink one is the free luteolin, and blue one is the polymer. Say for in case of TU212, this Nano-Luteolin, they have a IC50 of around 4, and luteolin, the free one, is a 6.9. This is a bit better. This one is a bit better, is higher luteolin. And in case of lung cancer cell, we found that both nano-luteolin and luteolin, they have comparable IC50 values. We also performed the colony formation assay, and using those nano-luteolin and luteolin both, and we saw that in case of head and neck cancer cell line, at a concentration of 5 micromolar, nano-luteolin can inhibit the cell growth, this colony formation a bit better than the free luteolin, but at a concentration of 10 micromolar, both of them can inhibit the colony formation. In case of lung cancer cell, we've observed that at a concentration of 5 micromolar, both nanoparticle and the free luteolin, they did the similar type of cell colony inhibition of colony formation, but at 10 micromolar concentration, nanoparticle did a better job of inhibiting the colony formation. So after this initial result of in vitro studies, we wanted to explore the polymers of different sizes to make nanoparticles of different size. So we used another polymer, but its a molecular weight is 3,000. The first time we used 10,000. So we made the nanoparticle, but in this time also we characterized it the same, but the nanoparticle size is much smaller, 15 nanometer. So initially we didn't have any idea which size is better for our purpose of cancer therapeutics. So you are trying many things at randomly. So we characterized similarly here also, but the size is smaller, this nanoparticle. And at that time we found a paper. So they have isolated and characterized a peptide which can target the EGFR, epidermal growth factor receptor. And this receptor is overexpressed in head and neck cancer cell and also in lung cancer cell. So we became enthusiastic to explore that how this EGFR targeting peptide, how it can target our nanoparticle for the cancer cell. So then we made this nanoparticle, and this time on the surface there is carboxylic acid group, the simple amide coupling. And this is the peptide, EGFR targeting peptide. And we formulated this nanoparticle on the surface with the peptide and characterized similarly by using the same techniques which I showed. So and initial study, this in vitro study, the cell growth inhibition assay, the SRB assay. So this one is this pointer is not very good. So this one, the blue colored one, is that nanoparticle on the surface, this EGFR peptide, and green color is the nanoparticle, and this is the free luteolin. So this one is lung cancer cell, H292, and this is head and neck cancer cell. So it seems from this in vitro assay that this peptide and nanoparticle conjugate, it can inhibit the cell growth a bit better than others, both in case of lung cancer cell line and head and neck cancer cell line. So at that point, we tried to explore the polymers of different molecular weights to get the nanoparticle of different sizes. After that, that time we had some resource constraint and time constraint. So we decided that we will not go for active targeting using the peptide. We'll go for the passive targeting. But we also wanted to understand that whether what is the uptake, cellular uptake of this nanoparticle, luteolin nanoparticle, which is synthesized. But that time we decided that we'll formulate a nanoparticle which will contain both the luteolin as well as a colored dye. So we took this sulforhodamine B dye and luteolin, the flavonoid compound, and formulated the nanoparticle. So this is the nanoparticle solution, and we characterized by the same techniques, the TEM. And after that, we did a study. We used the serum-starved head and neck cancer cell line, and we exposed these cells to the nanoparticle which is containing both the flavonoid anticancer compound as well as the sulforhodamine B. So this is the control, and this is the nanoparticle which is containing both the anticancer compound as well as the dye. So it seems that at different time points, with increasing time, there is more uptake of this nanoparticle. So since it's containing dye, we can see the color when we use the confocal images. In the next slide, so here we did this in vivo experiment using tumor xenograft mouse model, and we used that head and neck cancer cell line Tu212, and we developed the tumor here on the mouse. So there were 4 groups, and each group there were 7 mice, and there was on control. One group was given polymer, another group was given free luteolin, and another group was given this nanoparticle, Nano-Luteolin, we call it. So this is the tumor volume versus time. and the green color one is that Nano-Luteolin. And when we see this, this plot, this Nano-Luteolin showed more tumor growth inhibition. However, the p-value was significant only for two groups, comparing the two groups. Only p-value was significant when we compared the Nano-Luteolin with luteolin. Other values were, p-value was not very good. But to address this issue, so our next goal is that we have to increase the amount of luteolin encapsulation inside the nanoparticle. So that's what we have to work on, so that we can encapsulate more and can get more effect in reducing the tumor. We also did this, this Ki-67, which is a proliferation marker. So you see that Nano-Luteolin, there is less of proliferation, but compared to control and polymer and luteolin. So we are planning to do increase the encapsulation. And another thing can be done is that some other research groups, they are taking other naturally occurring compounds, like curcumin, and made analogs. And sometimes the analogs show better anti-cancer properties than the original compound found in nature. So we can eventually also do that type of medicinal chemistry approach, taking different analogs, and which will, if that shows better anti-cancer property. So these things need to be done in future. But I used this experience and tried to apply it to other projects. So here also another thing we did, we also checked in collaboration with another group. They have experience, they have expertise in prohibitin and phospho-prohibitin. So we did the Western blot, and prohibitin is a survival factor. So we have seen that this luteolin, it has inhibited the RAS, RAF, MEK, and ERK pathway. So that is another new finding when we saw the prohibitin level in these tumors. But this experience I used in another project. So we know that on the cell surface there are lots of carbohydrates and glycoproteins. And when any bacterial pathogen, they attack the host, so they bind to the receptors, the carbohydrates on the cell surface. And say when bacteria or virus attacks, so it binds to the cell through the cell surface carbohydrate. So in our company, we have a number of carbohydrates and analogs, which we have found that they have anti-infective properties. It can bind with virus and bacteria. So for example, we have one carbohydrate which can bind to the norovirus virus. So this is a CDC data that 49% of the intestinal diseases are caused by the norovirus. So when norovirus, so this is a norovirus, this is a virus-like particle, and just a computer modeling. So when it binds, this norovirus binds through this trisaccharide. So we have a carbohydrate compound which can bind moderately to the norovirus. So what we are doing is, so I have a polymeric carrier. So we have various glycans which can bind to the pathogen. So I have decorated them with different glycans. And each of glycan has shown to have anti-infectious properties from moderate to less. So we have synthesized this multivalent conjugation of glycan with biocompatible polymers, and the biological studies are going on. So hopefully we'll get some good result, and we'll share you in future in the next meeting. And these are the people who worked in this project. And thank you all for your time and listening the talk.
Thank you. Very nice talk. Very interesting. Any questions?
Hello, sir. That was a very nice talk. My doubt is nanoparticles ranging from 10 to 50 nanometers, don't they have easy chances of escaping the
renal
system?
Clearance.
Yeah, renal clearance.
Yeah, so that's why what we did, initially though we formulated the means 10 to 15 nanometer size nanoparticle, but we decided to use the 100 nanometer nanoparticle. So the biology we did, these in vivo studies with mice, we used the 100 nanometer size nanoparticle. But at the beginning, when we started the project, I didn't have any idea. So I was doing many things, trying, some naive approach at the beginning. But later I realized, and now I have a better understanding that which can give a better biological result. So maybe I have to do the whole project again.
And about the parameters, which parameter do you think is the most imperative one to get nanoparticles in the smaller size range? Like, is it the molecular weight of the polymer, or the stirring time, or sonication time period? Which one? Which parameter?
I didn't show, but both matter because the polymer size will affect the nanoparticle size. Because if you take a long polymer, then the nanoparticle will be big. But I didn't know at the beginning. So once I took a polymer, which is the PLA size was 70% and polylactic was 70% and PEG was 30%. And the nanoparticle we formulated, so it was very sticky. So when I was lyophilizing, I was not getting any powder. I was getting some, sticky like honey. If it keep you in the freezer, it becomes hard. So I could not solubilize it, the lyophilized sticky material. So I had to sonicate it and heat it and for a long time too. So those ratios of the hydrophobic polymer and hydrophilic polymer that affect the quality of the lyophilized powder and the solution. All these things matter.
Thank you. How this passive approach, these nanoparticles that are developed for passive transfer, do distinguish between the tumor cells and normal cells?
Oh, yeah. Actually, in case of passive approach, so the characteristics of the tumor vasculature is that it's leaky. So this nanoparticle, it does not have any targeting ligand. But since the vasculature is leaky, there is gap. So it can enter through this leaky vasculature into the tumor interstitium. And slowly, eventually, the polylactic acid, it can undergo hydrolysis. And this polymeric, the encapsulation, it will collapse and the drug will get released. So this is the passive targeting. But in case of active targeting, you have the ligand, so which can undergo the receptor-mediated endocytosis. So some pharmaceutical companies, since they are just doing passive targeting because they have got good result and they don't want to spend more money and they have already done the biology. So, for example, one company, you can check Cerulean in Boston. So they are doing the passive targeting. And some companies must be doing now all these antibody drug conjugate. But in our case, initially I tried this EGFR targeting peptide, but we had the time constraint and we had the funding issue also. So since it needs lots of peptides, so I was not making peptides, so we were buying from a company, so it's lots of money. So, and I wanted to finish the project quickly, so we decided we will do passive targeting, and we got a decent result, publishable. So, but you can do both. You can do many things.
So I was actually wondering why you specifically choose for Luteolin and not for any other flavonoid like Glycosyn or Genistin, which are also known to be anti-tumor active.
Yes, but Luteolin was shown to have anti-cancer activity against head and neck cancer cell. So our lab has a focus on head and neck cancer cell and lung cancer cell. So that's why. And we in our lab also there was another project where we used free Luteolin and we are doing some experiments. So you wanted to try to the same compound with nano so that we can compare the animal experiment result of the free compound, not nano, another animal experiment with nano. So there was some studies going on in the lab. So along the same line, we were trying this compound.
Any other questions? I have a short one. So you have a compound, IC50, about 5 micromolar. Is that good enough? Or if it's not, how do you improve it?
Yeah, actually, I should have added one summary slide. But we need to do more work because we need to increase the encapsulation efficiency of the nanoparticles. So if we can encapsulate more of this flavonoid compound, then we are hopeful that it will show more anti-cancer activity, and both in vitro and in vivo. Because in our lab there was an animal experiment in a different project where they gave the free compound oral gavage orally to the mice. But in that case, they put lots of compound inside the mice. So they showed better anti-tumor activity in vivo. But in my case, I encapsulated 3 times less than what they did free compound they put in mice. So we need to improve the efficiency of the encapsulation. That will help to solve this problem.
Thank you. Thank you for your wonderful talk.
Okay.